Hello.
Hello?
Hi, how are you?
Oh, fairly good.
Good.
Okay, so,
Okay.
well, we're waiting to see if we can make any progress on stability of tensegrity structures.
Right.
Be nice to throw one together and say, this is its stability.
Yeah. I haven't done that yet. But yeah. So, did you have anything more to say about that, or I haven't? I mean, I haven't really thought about the stability thing too much since we talked last. But
Okay, well, I guess others will come on. Well, we'll see if they do.
yeah, Hussein said he wanted to give an update, but I told him, I'd be available now. I don't know if he's going to show up though. Yeah.
Hello.
Hi. How are you?
What was the? Well, pretty good. I thought maybe this wasn't going to be on today, but
Oh well. Yeah.
well,
So
I just wanted the reference for that, and I'm sorry about the large dump of papers I gave you, but it's sort of what I've been looking at
when was it?
and not
Well,
doing much with.
when was that? That was last week or just now? Or
Yeah, last week.
oh, okay. Yeah.
Yeah, I think I sent them to you last Thursday.
Yeah. I'll go back and take a look. I don't know. Somehow I don't remember it, but maybe I do. And yeah, I've been busy.
Okay, yeah, well, do take a look at them because there's some that are biological and some that are tensegrity related.
Okay. Okay. Yeah, I think I remember now. Yeah. That's
Yeah,
fine.
you probably be more interested in the biological ones
Yeah. Yeah.
rather than the tensegrity ones.
Yeah.
I'm having trouble with the tensegrity math. I don't know what to study first. Like I need to study Lagrangian mechanics perhaps before understanding the math for tensegrity, although I don't think that's quite right. I need the prerequisites and I have no clue what they are.
Okay.
So if somebody could like give me a hint as to what the prerequisites are for the study of the tensegrity math, I sent you several papers on that. And if you could tell me what I need to study, then I could just go do it. And then maybe I'd understand what on earth they're talking about. My linear algebra does help, but it's more than linear algebra. This is
yeah.
some sort of complicated linear algebra.
Yeah. Yeah. We'll have to take a look. Yeah.
Yeah. Well, I'm fairly sure I have an understanding of math to at least an undergraduate level of electrical engineering. And this is like, I don't know.
Yeah, so
So
I'll take a
what
look
was
and
the
I'll tell you. Yeah, I'll tell you what the kind of math you need to know more of. I mean, it'd have to go through. I think we've gone through some of it, but I'll have to go back and make a list. So.
Okay, I really appreciate that.
Yeah.
Just knowing where I need to go with this. I'm sure I have the background math, but I need to know which direction to take it next to understand it.
Yeah. All right. So yeah, this.
I mean, this.
What was that?
Dr. Sharif did take one look at it, and he said, "If you expect me to study that, I don't have time, so I'm not doing it." I'm going, you don't like this guy is base is math.
Okay.
An electrical engineering professor whose base is mathematics.
Yeah.
That's how he does all his projects.
Yeah.
And it's just like, and he didn't know it well enough.
Yeah, it's very specific to the problem. Yeah. Okay, welcome to the final DevoWorm of the year. A couple things I'd like to go over for the end of the year. The first thing I'd like to talk about is that we had our annual meeting last week, last Tuesday, the OpenWorm Annual Meeting. And I gave a report on DevoWorm, and we also talked a little bit about some of the things going on in OpenWorm. One of the big things is that people are within the project are using vibe coding to sort of get some older things working. So one of the old things that we had that disappeared over the years was the iPhone app. There was an iPhone app where they did things with the worm simulation. And so that's something they're trying to revive. Other legacy pieces of software that they're trying to modernize and revive as well. So, whatever your feelings about vibe coding are, that is still a pretty useful thing to get old things working again, especially in open source. I think that's a very good development. So that's what's going on there. And they're doing a bunch of other things that I don't really want to get into right now. But suffice it to say that, OpenWorm is going strong. Now, as for our work, I developed a talk for the annual report. This is the talk. It's on our YouTube channel, so it's about 41 minutes long, and you can go through and look at all the things that we've done this year. So we didn't do as many publications as I had would have hoped, but still, we did a lot of topical deep dives, and we've been doing research, and hopefully in 2026 we can get some of that published. This is one example of something that was talked about in the report. This is our work on multiphysics and tensegrity. So this year we talked a lot about multiphysics models. We talked about models in which you have multiple physical processes going on simultaneously. So in the embryo, you might have flows, liquid flows. You might have structural issues. You might have, in this case, it says thermal, but it's really like, say, metabolism. And then you have in this case, it says electromagnetism. You might have bioelectricity. So those are all going on at the same time in developing cells and developing connectomes. And so this is a very interesting way to approach embryogenesis. But the problem is, of course, is that we can't necessarily model biological systems using conventional multiphysics software. So there are a couple things that are the problem there. The first is that, we want to be able to model biological processes, birth-death processes, differentiation processes, division. So a lot of multiphysics software deals with mechanical systems or, basically things that are non-living or don't have the traits that we want to incorporate from living systems. Another problem, of course, is that a lot of the super stable structures that we want to model that represent complex tissue dynamics are indeterminate structures in the eyes of the software. So this is where we have multiple sources of, say, like compression that balance out, and there's a mathematical inconsistency that won't allow for the proper modeling of the dynamics. You end up with models that fail immediately, and of course, obviously, that's not useful to us. So we need to think about multiphysics, we need to think about this indeterminacy problem, and then we need to think about how tensegrities play a role in stable biological structures, going from something that's undifferentiated to something that is stable and robust. So that's a little bit about the annual report. You can go onto the YouTube channel and check that out. And hopefully that gives us ideas about what we might want to do for next year. So thinking about what we want to do for next year, obviously we want to put some more publications out into the world. Also, maybe if you're interested in presenting at one of our meetings, please contact myself, and you'll be able to do that if you would like. We also have opportunities with Google Summer of Code. We have opportunities to work with OpenWorm. They have an opportunity called a studentship, which is where they, you usually have to be some sort of student, either a graduate student or an undergraduate student. You have to have an interest in working with biological data, And you have to have some sort of really focused project that is portable to this studentship. But the benefit of that, of course, is that you work with OpenWorm and you develop something useful. So it might be an opportunity for some of you. For others of you, if you're interested in doing research, if you're interested in building projects out, you might do that in conjunction with your coursework or with something that you want to accomplish in your professional career. That's something else we can do as well. We have, if you go through our YouTube channel, there are a number of topics that might be of interest to people, not just the tensegrity work, but we also have things having to do with biological questions. We're looking at syncytium and C. elegans, which are cells that have multiple nuclei. We're looking at that in conjunction with the lineage tree. We also have applied machine learning projects, largely these days focused around graph neural networks, but also a lot of machine learning oriented towards development, and other types of mathematical modeling oriented towards development as well. To finish off the year, I wanted to talk about a couple of papers. This is the first paper I want to talk about. So this is a bioRxiv preprint, and the name of this paper is Living Neurosheets, Engineering Readily Deployable Neural Architectures. And this is from a number of authors. John Beggs, who does a lot of things with neural criticality, Attiya Gazzola, who's at UIUC, Tahir Saif, who's at UIUC, and so there are a lot of authors on this paper. And so this looks like an interesting sort of bio-inspired architecture for, sort of maybe as a replacement for neural networks, but definitely as a neural control mechanism. So why don't we go through this abstract? Motivated by the brain's extraordinary processing abilities, the promise of queryable neural models, and the potential for biohybrid computing, we present living neurosheets, practical, malleable, and modular platforms for the realization of complex in vitro living neural architectures, bio-inspired or beyond evolutionary precedent. By innovating porous sheets, substrates employed for millennia in the form of paper are here reimagined, inheriting neural function and exceptional versatility. Through pre-programmed porosity, deposited neurons are held in place for durable and reproducible spatial control. By cutting, folding, and stacking, elaborate neural topologies can be designed, batch fabricated, and modularly composed. In combination with tissue-matched optoelectronics, neurosheets deliver a powerful foundation for the multimodal integration of neural motifs. This is illustrated via planar tissues, gyri-inspired manifolds, and laminar cortical mimics, where we demonstrate control over entrainment, synchronization, propagation, suppression, and augmentation dynamics, recapitulating fundamental building blocks of in vivo neural activity. So this is, what they're doing here is they're creating a material, and they're building in these features of basically a network, and then they're building higher order structures with them. So if you know, of course, about work on gyri and sulci in the human brain and the surface of the human brain, where the human brain, the neocortex, has expanded to the point where it's so large that it needs to fold, and it folds up quite intricately to fit into the brain case. And those folds, sort of their undulations, are known as gyri, or gyri and sulci. And so these folds allow for this huge sheet to fit within the brain case. But then there are other features of these folds that produce a certain topological complexity. And so this is something that I guess they're addressing here by allowing us to build these sheets that have some sort of neural control function, and then configuring the sheets in different ways. You can fold them, you can stack them, you can cut them and form different shapes depending on how they would be implemented. So this is kind of an interesting advance from the standpoint of the physical aspect of networks. How do you put networks sort of on a material. And then, of course, we've talked about neural organoids, where you can grow up structures, you can grow up culture, three-dimensional structures of fairly high complexity. But we haven't really talked about sculpting those neural organoids into highly modular and configurable sheets. So that's what they're doing here. So they start out and say, what if living neural systems could be engineered as programmable circuits with freedom to prescribe architectures, guide information flow, and enable rich multimodal interaction? No longer constrained by the complexity of the intact brain, neural topologies could be synthesized to recapitulate hallmarks of sensory processing, perception, or learning, advancing fundamental understanding, and promoting biohybrid technologies that harness life for computation. So this is what they're calling in vitro neuroscience, as along with your neural organoids and many other in vitro approaches. While in vitro neuroscience has long hinted at this transformative potential, existing technologies fall short of delivering engineering-level control over neural design, scalability, reproducibility, and simplicity. Key requirements are efficient prototyping, systematic interrogation, and widespread adoption. So here they get into brain organoids, and they compare what they're doing with brain organoids. So brain organoids, powerful developmental and disease models, offer limited structural control and reproducibility. Bioprinting, which is their alternative, so they're printing structures onto sheets and then reconfiguring them, provides versatility. However, neural applications remain circumscribed, hampered by material constraints. In both cases, challenges tied to cell migration and structural integrity persist. Left unchecked, neural migration, a hallmark of development, indeed disrupts mature architectures where structural consistency is instead critical for sustaining reliable function over time. So on these sheets, of course, they don't have any developmental aspect. So it's just you have to know what you want to fabricate, you fabricate it, you build the structure, and there you go. In brain organoids, and of course in the brain itself, we have these developmental processes. So we have neural migration, and of course these are things that can sort of self-organize or configure these types of systems, brain organoids, or natural developmental neural systems. And these are, of course, guided by cues that are going to lead to some known functional circuit. So you're dealing with something that if you know exactly what you want to fabricate, this the Neurosheets is sort of better. But if you want to self-organize a circuit, or if you want to build upon the self-organizing potential of an organism, then you need things like neural migration. And in fact, this would disrupt what was going on in the Neurosheet. So that's kind of what they're getting at with this comparison between the technologies. Now they mention that both technologies suffer from high barriers to entry. So consequently, in vitro studies of neural processing continue to rely on 2D cultures. So, we have two-dimensional cultures where we can record activity. We can grow those pretty well, different cell types, or, we can reprogram cells to neural cells, or we can work with neural precursors or neural stem cells. But, of course, then when you move to the third dimension, that's when you start to get problems, in terms of scaling things up. And that's what they're getting at here. So 2D cultures are prized for their simplicity, but they're unable to replicate the hierarchical complexity of native neural tissue. So just having that two-dimensional, that sheet of cells, you can't replicate that complexity. You just have a bunch of cells in a dish that maybe have some spatial complexity, but not any hierarchical complexity that is of note. So in this context, we introduce living Neurosheets, malleable biohybrid platforms that enable neural design freedom, streamlined fabrication, and robust interrogation. porous sheets, exceptionally versatile materials with a millennial history of diverse applications, are cut to design, innervated, folded, and modularly assembled before placement onto optoelectronic interfaces. So they're using this in conjunction with a lot of the optoelectronic technologies that we have, so we can stimulate them and record from them and have this as sort of a closed loop system. This workflow is intuitive as handling a sheet of paper, offers scalable control, and enables deployment for reproducible experimentation within seconds. So they mentioned, how do you print neurons onto a sheet in the first place? So importantly, neuronal bodies are held in place through pre-programmed porosity, which prevents cell migration and supports the formation of robust macroarchitectures, while also allowing neural projections to traverse the pores and establish interconnected 3D networks. So you have these sheets where you have the neuronal bodies that they sit in sort of their well. Then they can make connections with other neurons in a three-dimensional matrix. You want to prevent cell migration, so if you have sort of a design for a circuit, you can print out that circuit, you can put the neurons, seed it with neurons, and then you have your network. And the neurons can't move around, although, maybe that's something that would yield an interesting result. We're interested in control here, so we want to keep those things constant. From a fundamental standpoint, as synaptic connectivity, or what they term the microarchitecture, is continuously remodeled by plasticity and maturation, the control afforded by neurosheets over macro-scale network organization allows us to decouple structural from functional change. And this, of course, is important for different types of neuroscience investigation and computation. The reversible integration with optoelectronics then permits us to systematically probe our architectural motifs, shape emergent neural dynamics. So this paper I'm talking about is this Living Neurosheets, Engineering Readily Deployable Neural Architectures. This is from the bioRxiv. It was published, I think, last week or a couple weeks ago. So this is, they're talking about these different types of, I guess they're doing bioprinting. And this is actually something Morgan is interested in. So, printing out neurosheets, which have like neurons on them, and then they can build, they can print the sheets out and then configure them into three-dimensional structures. So they can have the neurons on the sheets, configure the sheets in different ways, and the neurons can connect to form this three-dimensional structure.
And they're saying migration is important to building brain structure. Yes.
Yes. Yeah, it's well, they're saying that they can't have migration in these sheets, but you can also, what they're interested in is the synaptic connectivity. So it's like the neurons will send out axonal processes, and then when they meet other axonal processes, they build synapses, and the synapses enable the connection between the cells. So, although you could have a developmental structure like, what you see in something like a neural organoid, and you get a lot of migration there, and you get all these interesting structures that are sort of like what you see in a developing brain. But this is kind of where you know exactly what kind of network you want. You print it out, and you can configure it. That's, I guess, the
okay.
So yeah, they're kind of thinking about planar neural tissues of varying topology, and this allows us maybe to study things like information encoding, entrainment, synchronization, and propagation. So these are hallmarks of in vivo neural activity found in sensory processing, memory, and recall. So you have these kind of test beds for looking at these very simple problems, or what would become simple problems of information encoding. And then drawing on this, we design networks that autonomously enhance and suppress encoded information, illustrating the importance of macro-scale topology in orchestrating emergent neural dynamics. By expanding the design space through mechanical buckling and folding, so again, we have this aspect of folding and buckling like we see in the neocortical sheets in human brain development. where you have a sheet that's so large it has to buckle to fit into the brain case. In this case, you might want to fit your large sheet into like a small space, like a robotic um, vehicle, or maybe something like, if you want to do some sort of transplantation in humans to replace a piece of brain tissue, or have a small restricted space where you need to fit this tissue, you fold it up and get this kind of, this allows for that.
Okay, but there's layers in the brain, and
Yeah.
they're developed from the neurons growing in 3D.
Yeah, they do configure themselves in 3D, but the idea is that they kind of have these connections that are vertical as well as horizontal, so that things go up and down these columns. And so you have a bunch of cortical columns, and then you have the sheet, which is a series of cortical columns in parallel, and then that sheet folds up. So it's like this really complex topology of, and then the information processing happens both vertically and horizontally. So vertically up and down
Yeah.
a sheet, horizontally across columns. So, that's kind of what the people doing hierarchical temporal modeling are doing. They're looking at these cortical columns and using that as the unit of computation. So that'd be interesting to see like hierarchical temporal modeling people get into this paper. See what they think about it. But I'm not I know something about hierarchical temporal modeling, but I couldn't give you the critique that's needed here.
Oh, okay.
Yeah. By expanding the design space, they realize motifs inspired by gyri and sulci, structures present in the cortical lamina associated with higher order cognitive functions. So this is where they're getting into this analogy with the neocortical folding. These compliant neural manifolds can serve as models to investigate the role of curved packings in processing, or can be deployed in deformable robots for embedded neuromuscular control. And then lastly, through modular stacking, we build reductionist cortical mimics that are top-bottom electrically interfaced, demonstrating activity propagation and synchronization in laminar constructs. So this is where they're talking about top-bottom, meaning they have these cortical columns that are, but in this case, what they're trying to do is just have like places where you can access this information processing structure. So if you have a robot, you have some sensory input, you have these layers, and then you have the output to the effector, something like that. And so that's what they're ending in. So this is an example of their Neurosheet. This is fabrication and structural characterization. So this is A here is the laser cutting, the fabrication process. They have this laser cutting of filter paper. Then they build these features into the surface. They have this fiber network. That's the design. Then the innervation in B, you have PDMS substrate. You suspend the cells, and then you have this, then you grow them in this medium. 3 is the fluorescent imaging. So once you have the design, and then you have the fabrication, and you get the neurons in these structures, you want to confirm to see if the neurons actually are in the structure, and we can see that the neurons are fluorescing properly, and that you can define the edge of the sheet so you can find exactly where they've ended up and confirm that. D is characterization, where you have this is electron microscopy. So we have the neural cells and the fibers. We can see those in the electron microscopy on the feature. You have neural cells here, and so this bar is 30 microns, so we're dealing with a pretty small structure. It's not quite a nanostructure, but it's a microstructure, and so you have these different configurations of the neural cells. This is the cell side, where the cells are they're placed in these wells, and then they're innervating the structure, and they're making connections. So you can see that you have the cell bodies that are separate but they're making connections between each other. The opposing side, we have no cells on that side, so we only have the cells on the side we want, so we can have greater control. The opposing side is where we see things like cellulose acetate and neurites, so things that are maybe supporting that substrate of the substrate and then the neurons on the other side. This is the immunostaining, so we can see what, that the cells are doing things, that they're expressing different things. And then this is batch fabrication, and that's kind of where they're going across scales, where they're doing these different sheets in parallel. So that's what you can see here. Okay, so that's just kind of an example of what they're doing. If you're interested in the microstructures and the fabrication, basically they begin by selecting biocompatible materials at pore sizes tailored to simultaneously confine cell somas, enable efficient hydrogel infusion, and support neurite extension. So they have to pick sort of biocompatible materials that allow for the compliance needed for cells to sort of find a place to settle, establish their axonal processes, and survive in that environment for any period of time. So that's what they're trying to do there. And so they need to be able to have a material that supports neurite extension both laterally and orthogonally throughout the sheet volume. And so what they want to eventually do is achieve three-dimensional connectivity. And they're trying to, of course, mimic the radial interfacing of the allocortex and neocortex. And so again, they're mimicking the neocortex in these cortical columns. With simplicity and broad accessibility in mind, we consider two cost-effective and widely available solutions. The first is fiber-reinforced cellulose acetate filters. And then we have tracked etched polycarbonate filters with pore diameters ranging from 0.8 to 20 microns and thicknesses ranging from 32 to 135 microns. So they eventually picked the 100 micron thick cellulose acetate filters with a 5 micron pore size. The application using 32 micron thick polycarbonate filters with 20 micron pores is highlighted in figure 5 as an alternative. And so they just kind of try to characterize this as much as possible to see what's optimal. The treated constructs are then placed on a hydrophobic PDMS substrate to establish a wettability contrast. This contrast enables precise confinement of liquids within the seeding area, causing droplets to bead up, thus ensuring accurate control over the deposited hydrogel volume and cell density. So they use the substrate material to control the cells to make sure they don't migrate away. In this case, we don't want migration. We want something that will be maintained in sort of these beads. So we don't want to necessarily have, we want to have this hydrophobic surface so that we can have beading, so that we can constrain where the cell body is. And so that's what they're doing here. Next, collagen matrigel hydrogel is infused to provide defined mechanical and biochemical cues that support the maturation of neuronal networks. The resulting sheet hydrogel constructs, which will serve as tissue culture substrates, will be batch fabricated, characterized, and stored. Modularity also permits tapping into the existing infrastructure for sheet functionalization, for microfluidic integration to flexible electronic patterning. So yeah, then they talk about how they raise their neurons, how they get those the neurons to survive, and then they talk a little bit about how they confirm that the networks are active and that the cells are on the substrate and things like that. Then they do, of course, optoelectronic interfacing. So they want to be able to record and stimulate the cells as well as image them. So this is functional characterization. This is the Neurosheet in a sort of a cell culture container. This is the MIV platform, and you can see that they're recording from this micro electrode array, 512 channels, and you have the channels over time. So actually, we can find not only that we can characterize not only that the cells are in place and that they're active through the stains that we saw or the fluorescence that we saw, but we can also do electrical recordings, and we find that the neurons produce these global and spatially localized bursts. So if you know, I noticed that John Beggs is on this paper, so of course he does the things with neural avalanches. It's been his kind of thing that he's done over the years, neural criticality, neural avalanches. And so one of the things that happens in neural cultures or in neuro groups, populations of neurons, is that they produce these bursts that are sort of exhibit this sort of self-organized criticality. That is, they produce these bursts that conform to a power law over time. So you get these bursts that are irregular in their time interval, and you get these in a neural population. So you get a bunch of neurons that sort of exhibit bursts, at these irregular points in time. And the idea is that they're related to all sorts of different collective phenomena. And so you see this across the culture, across the sheet in this case, but you also see this localized. So you see sometimes a few neurons, or maybe even a single neuron, will exhibit a burst. And burstiness is related to how the cells are connected and how they're sort of exchanging information. So the thing that you need to know from this is that their exchange of information is nonlinear, and so it results in these bursts. So you see that, from some of the channels you get like these localized bursts over maybe a few channels, whereas you get this global burst where almost all of the channels are exhibiting this burstiness at one time. And so you see this, of course, because
what frequency? Sorry, what frequency is the bursting?
I'm not sure. Let's see if they have it in here.
I guess you could calculate it.
Yeah. I mean, they don't really get into that here. Yeah. But
7, 8, 10. So once every 6 seconds or something.
Yeah, roughly. I mean, it's not like there's an average to it, but it's it doesn't conform exactly to the average. It's kind of the idea.
A cure for or a partial cure for Alzheimer's is 40 hertz stimulation of the brain with electrodes. Yeah, it's some pioneering work at the University of Manitoba.
Okay.
And it slows down Alzheimer's. It helps keep them functional longer.
Yeah.
It improves their functioning.
Yeah.
Anyway, it was interesting. It was on YouTube. They put the conference on YouTube.
Okay.
I could also get you that if you're interested. But it just the 40 hertz seemed to be what they chose for stimulation frequency.
Right. There's this old idea of binding, which is where there's this global signal. I think it's at around 40 hertz, where different parts of the brain are all kind of synchronized in that frequency band, the activity. And so then the idea was that that's sort of a global signal of consciousness or something. And I don't know, that's a literature that you don't really want to go too deep into. But it's that was an idea people had at one time, and it's like an aspect of the EEG spectrum that's really interesting to people. So.
Yeah, I got to see this is Dr. Mosavi, University of Manitoba. I got to see her equipment. I tried it out on my arm, and it made my arm tingly. Just,
Yeah.
but yeah, she attaches electrodes to the forehead and has tried it out on older population who has Alzheimer's, and it does improve cognitive functioning.
Yeah.
Anyway.
Yeah. Okay. So yeah, so they're able to reproduce these bursts, which you see in, in actual brains. So that's good. And you also see this in microelectrode array recordings of two-dimensional cultures. So that's also good. Then we have this optical stimulation. So you have a difference between sort of a stimulation of the culture. And so if you're familiar with optogenetics, they usually use some sort of genetic marker to make the cell responsive to light. And then they provide a light flash, and then the cells respond to the light. So you have this light source, you get this stimulation, and then you get this response afterwards. So that's what they're doing with the optical stimulation. And people do this oftentimes. This method is, they've done this within living in vivo brains where they have, like they've done this in mice where they stimulate part of the cortex. They can basically turn it on and off with this optical stimulation. And so they can get greater control over different types of, say, like motor dysfunctions or other types of dysfunctions in the brain, seizures and so forth. You stimulate the cells with light, or you basically entrain them to a light source, and they can, you get these types of results. So that's what they're doing here. Then they look at the normalized firing rate. They're looking at sort of this adaptation curve where there's consistent response after reuse. We have electrical stimulation, so we want to know if like stimulation is changing the activity of the neuronal culture as opposed to just being a random thing that's thrown in there. If you have a light source and you're kind of stimulating cells, you can do that, and it's it could just be a random phenomena. So if they're not connected in any particular way, or if they're not connected at all, they can respond to the light source, and it's all random, and that doesn't really mimic the brain, or it doesn't really do anything that the brain would do. In this case, what they're doing is they're verifying that there's some stimulated aspect to the firing rate, that there are patterns of correlation there. All right. Then they do some calcium imaging. They have this calcium indicator and this intensity curve over time. So they can characterize the bursting by the intensity of this calcium indicator over time. They actually have for 50,000 cells versus 150,000 cells. So I guess this suggests that there's some sort of density dependence here. That there's, I guess basically they say here, primary cortical neurons exhibit density-dependent dynamics, lower density showing mostly asynchronous activity, and higher densities producing periodic synchronized responses. That's what they've got here. So and then of course they talk about planar tissue topologies. So this is where we're looking at the geometries of neural dynamics and control. They refer to a directed emergence of intractably complex microscopic connectivity via well-defined macroscopic boundary conditions. And of course, this is something that's needed, is this directed emergence, meaning that you can have a network that wires itself sort of spontaneously, and like we talked about up here, you can have this spontaneous response, and that's not necessarily very useful. Now, in an in vivo context, you have chemical cues. We have the substrate that is sort of, formed. We have cell migration and this matrix that's formed in development that enables sort of this directed emergence in biological systems. So what they're trying to do is mimic sort of what development provides to an emerging brain or an emerging connectome in an in vitro setting. And so this is, of course, a problem with neural organoids, trying to get useful collections of tissues out of basically putting a bunch of cells into a culture and hoping that they sort of self-organize in the right way. And so that's kind of what they're getting at with directed emergence. And so they're looking at different ways to do this topologically as well. So to impart dynamics of transport, directionality, and integration, we move beyond genus 0 typologies, which are square tissues, and adopt genus 1 configurations, enabling branching and merging. So this is actually, I guess, mathematical topology. I'm not familiar with these different genera of topologies, but this is, again, moving from the square tissue to something with some biological-like structure. So then we begin with a ring motif, which is genus 1 topology, characterized by a periodic boundary condition that allows inputs to diverge, converge, and self-interact. We'll see in figure 3 what that looks like. Ring-like motifs are common across neural systems, from the fly central complex, encoding head direction, to cnidarian nerve nets around the oral or bell margins of jellyfish. So these are things that coordinate feeding and locomotion. So what they're trying to do is they're trying to mimic different types of ring-like motifs of cells in biological systems. We have the fly central complex, we have the cnidarian nerve nets, we also have the C. elegans ring, the nerve ring, and C. elegans, but they don't mention it here. So basically, we have this common topology that we see in different organisms. So that's what they're trying to do with these sheets. Ring topologies are then cut, treated, and innervated with either wild type or transfected motor neurons, guided by preliminary observations of rectangular strip systems. The ring's outer and inner diameters are set to 4 millimeters and 10 millimeters respectively. This is large enough to prevent spontaneous global synchronization and enable long-range activity propagation, yet small enough to limit dissipation and allow dynamics to spatially interact. So basically, they want to prevent sort of synchronization by means of being like too small. So, you can have, like, if you have a very small area, cells can spontaneously sort of synchronize their activity, and they really kind of interact globally, and that isn't really the kind of directed emergence that we want. We want this system to be large enough to sort of represent a larger area of space, but also synchronized through a combination of local and global connections. And so that's what they're doing here. They're just trying to find the optimal size for this structure, but they don't want to make them too big so that they can't have cells spatially interact and dissipate into something that isn't self-organized. So there's this trade-off, and again, I don't know if this is a function of the technology or a function of the biology, but they try to find the sweet spot in between them. And so to enable precise electrophysiological monitoring and manipulation, custom ring-shaped high-density microelectrode arrays are fabricated and matched to the tissue. So they hypothesize that toroidal tissues, which are these rings, or these donuts of tissue, which can effectively be thought of as a periodic axisymmetric branch. So this is where, these toroids are basically branches, and they're sort of periodic. Will support bifurcation dynamics, parallel transport, and two-stream interaction. Such systems thus represent minimal models for investigating mechanisms of signal copy, redundancy, propagation, or refractory period localization, all mechanisms biologically and computationally relevant. So this is figure 3. These are these planar tissue topologies. So you have up at the top, you have the ring tissues. So you have these rings, they're basically like donuts. And remember, they're three-dimensional. And so you have this ring that's the toroid. You characterize this by looking at the DAPI stain. You're looking at the stain that confirms the neuronal bodies. And then you're looking at a stain that confirms the astrocytes. So you have these three stains that you need to do to verify localization of neurons and supporting tissues and all of that. Then they have their microelectrodes in here and the ring tissue. You can just, they've characterized all of that. Then they characterize the electrophysiology in 3. And in 4, we have this optical stimulation. We have this bifurcation and self-copying. We have these ripples that occur in the activity, this radial average. We have propagation along the ring. And then this self-suppression because of the refractory period. So basically, we have the stimulation at one end of the torus. It goes in both directions, and it hits the other side, and there's this refractory period once it hits the other side where there's self-suppression. So if you make these too small, you get this self-suppression really early, and it doesn't allow for proper propagation. But if you make them too large, the propagation is way too long. And it, so that there's this happy medium here that you have that you need to achieve. And so they have these are, I guess, spatial recordings. So you see the strength of the signal in hertz. So you see that the signal propagates outward and it decays over time. So it's it's not like it doesn't just kind of go as far as it wants. It kind of dies out at the original source of stimulation and continues on through the torus. So this is all to say that the geometry is very important. It's not only important for applications, but it's important to get the dynamics right on these topologies, because after all, neural tissues aren't just networks, they're networks on some sort of topology. This is a Y tissue construct where they have this branching demonstrated. So they have the bottom branch, the left and right branch. Again, they're characterizing the structure of the DAPI stain, the neurons, and the astrocytes. Then you have this structure where you have a stimulation site at either end of either branch. You have a recording site down at the base of either branch and in the bottom branch. And so then they're looking at these bands, these EEG bands. So this is a 7 hertz theta band signal. This is a 3 hertz delta band signal. And then they're looking at concurrent stimulation on each branch. So in this case, on the right branch, we have the 7 hertz theta, which is stimulating from the right. You see that most of the activity is on the right branch. The stimulation at 3 hertz of delta, which is stimulation from the left branch. So you see that they basically remain in their branches and maybe propagate out into the other branches a little bit. But then when you get this concurrent stimulation at 3 hertz delta and 7 hertz theta, you get this 1 hertz slow delta that occurs in the bottom branch. So that's an interesting point that they're making here. And so this is, where they're doing this, they're looking at mean firing rate activity after stimulation, and they're looking at where this is encoding in these different neurons. So this is what they call signal augmentation down on the bottom branch, where you have this concurrent stimulation coming down both branches and then combining in the bottom branch. It's sort of this sort of directed emergent property. Okay. And then of course they talk about tissue manifolds. So having established planar spatial programmability, we next extend design space through folding and buckling to create functionally innervated 3-dimensional manifolds, which are these compliant neural structures unattainable with current approaches. So being able to bend and fold them into spaces and having them sort of be these analogies of the gyri and sulci of human brains. So this is a way we can achieve dense packing and support rich connectivity. And so from a biohybrid robotic perspective, integrating nervous systems into deformable motile platforms demands neural architectures that conform to dynamic body shapes while maintaining functional integrity. So actually, this is something that we talked about in, this is a while back in our Saturday morning NeuroSim meeting, where we talked about these structures that they used for designing neural networks on substrates, where they used origami and kirigami. I think Morgan might remember this paper, but it's basically where they take sheets and they fold them up. If you're familiar with origami, it's folding up the sheet in a certain way so that you can build a complex structure. In the case of origami, you could take a flat sheet of paper, fold it up in the right way and form like a swan, or form some sort of structure that is quite complex just from a flat sheet of paper. Kirigami is a little bit different, but it's basically a very similar technique, and you can design different things from a flat surface in that way as well. So that's kind of what they're.
The kirigami is it's the sections of it are joined by triangles, small triangles.
Okay.
And that's why I thought it was similar to a cell structure with the triangular junctions.
Yeah.
So
trijunctions.
Yeah.
Yeah.
Anyway, they use it for molecular structures quite often.
Yeah.
And you can form some interesting metamaterials using that idea.
Right. Yeah, this is something people are experimenting with. Where
Okay.
you can reconfigure the topologies. You can take a sheet and fold it in different ways. So that's kind of what they're trying to do here. And yeah, there's a whole literature on it. I'm not sure if people are doing it to look directly at like trijunctions or cellular structures, but that'd actually be interesting. Especially in the context of like tensegrities and other things like that.
Yeah, it just was an interesting idea.
Yeah.
I tried to make a 2D cellular structure, and it was good. It kind of worked, but I didn't pursue it because it would have taken extra time.
Yeah.
But I didn't have time. So, yeah, and I put something in the chat. That's the YouTube channel where Dr. Masavi presented some of her research. She actually got her grad students to make a program where you could drive as an older adult as part of keeping your brain sharp, and that's part of this. You do the brain stimulation, and then you have homework to do, and part of it's trying to drive with the simulation program and find your way through a house, and there's a bunch of games for Alzheimer's patients that she's made.
Yeah. Good.
So it's pretty interesting research. If my brain was going, I think I'd prefer to spend my time doing games on the computer, like driving, rather than just sitting staring at a wall. So
that's what they recommend.
So I think it's great. Yeah. Yes. So the stuff she's come up with is very good.
Yeah. So let me finish up the paper. Just this last part. I think it's probably So they're doing origami and kirigami, and it's interesting that they're actually building now these folded structures, and this is where they're kind of building these structures with neurons. They're recording from them, and they're finding out that they're able to replicate these avalanches, as we talked about, where you have these bursts, the spontaneous burstiness. Sometimes the bursts are very small, sometimes they're very large and global, and you're able to detect these in the structures. And so we're actually able to realize some neural control in these folded structures. So that's kind of what they're getting at with this. And then the next step, of course, is to make laminar tissues or laminar circuits that have layers. And so then we're getting into these cortical columns and other things like that. So one of the reasons I wanted to talk about this paper as well is because it's really interesting, and it does tie in a lot with what Morgan is trying to do and one of his interests. So he's interested in bioprinting and sort of these hybrid 3D structures. But he's been talking about it, of course, in terms of neural organoids or assembloids. And we talked about what those are, which are these three-dimensional cultures where you seed it with a bunch of precursor cells or stem cells. They differentiate in vitro. You give it the right chemical composition, and it will produce, hopefully, structures that, maybe they're layered structures or multicellular structures. You're getting basically a sort of a pseudo embryo where you're getting different structures or different types of cells that are functionally independent and interdependent. So they're kind of forming circuits. But at that level of control, it's hard to maintain a level of control over those. And, then he has this aspect of bioprinting. So printing them out on sheets and depositing them in sort of a more desirable set of topologies and things like that. So I'm hoping that Morgan takes a look at this and gives some feedback on what he thinks about the technology, because I think it's really interesting.
Yeah, don't forget about the mechanical stimulation.
Oh yeah, definitely. And they don't, they talk about the optical stimulation, but of course, when you have these structures, there is the mechanical aspect. Of course, they constrain migration of cells, but there are these other, so I don't think they actually make contact in that way, but they're actually sending their processes out into this structure. And so they're interacting in ways that, might be very interesting, especially in the organoids. We can't control this, so we have a lot of mechanical interactions there.
I think they need to pay attention to how they're being mechanically manipulated as well. Like the stress and strains in the gel are important as well.
Yeah.
If they don't know how come the results worked here and they didn't work over there, look at the mechanics.
Yeah.
Yeah. So it's an important aspect of that, and they need to pay attention. That's all.
Okay. Yeah.
Yeah, but it looks like, some of these structures are kind of tensegrity structures too, perhaps.
Yeah.
Like they're in tension, or they should be in tension in order to operate correctly. Anyways.
Well, that's all for today. Yeah, thanks for.
Okay, just saying our cells are in tension in our body.
Definitely. Yeah.
It just is. Yeah. Anyways, do take a look at the YouTube video. You'll find it interesting, I'm sure.
Yeah. And have a good holiday. And
okay.
Yeah. See you in the new year.
Okay. All right. Yes. Have a good holiday.
You too.
Bye.